Welcome to the Deep Dive. Today, we're diving into some really fascinating brand new genetic research.
Yeah, this is hot off the press.
It is. We've got a Nature Genetics paper just out April 2025. And our mission really is to unpack the key findings about a newly discovered neurodevelopmental disorder. What's the big picture here? What does it tell us?
Well, what's really exciting about this one is we're right at the edge of discovery. This paper, it pinpoints mutations in a specific gene, RNU2-2.
Okay.
And these mutations seem to cause a pretty severe neurodevelopmental disorder. But the really big headline here isn't just, new gene linked to disorder.
Right.
It's that this gene, RNU2-2, was one we basically thought was genetic junk, a pseudogene.
A pseudogene, so like non-functional relic.
Exactly. RNU2T, they called it. We thought it was just sitting there not doing much. And now it turns out it's a critical player.
Yeah.
That really flips things on their head a bit, doesn't it?
It really does. A gene we were ignoring turns out to be crucial for a serious condition. That's quite a twist. And it's not totally out of the blue, is it? I remember reading there's a connection to earlier work.
Absolutely, yeah. This builds directly on research that found mutations in a similar gene, RNU2-2.
Ah, that causes ReNU Syndrome.
That's the one.
Yeah.
A more common neurodevelopmental disorder. And what's interesting is the researchers estimate this new RNU2-2 disorder is about maybe 20% as prevalent as ReNU syndrome.
20%. That's still quite significant. It's not vanishingly rare then.
Not at all. It clearly represents a substantial number of individuals affected by these kinds of conditions.
Okay, so both RNU2-2 and RNU4-2, they're involved in some really core cellular processes, I gather. Something about snRNAs and the spliceosome. Sounds complicated. Why is that important?
Right. It does sound deep in the weeds, but it's fundamental. Both genes make small nuclear RNAs, snRNAs. RNU2-2 specifically makes one called U2-2.
U2-2. Got it.
And these snRNAs are essential cogs in the spliceosome machinery. Think of the spliceosome as like the cell's film editor for our genes.
Okay, I like that analogy.
It takes the raw footage, the pre-mRNA, which has extra bits called introns.
The parts you see on the cutting room floor.
Exactly. And the spliceosome, powered by snRNAs like U2-2, precisely cuts out those introns to create the final movie, the mature mRNA, that actually tells the cell how to build a protein.
And most of our genes need this editing process.
The vast majority, yes. So this discovery really hammers home how vital these non-coding genes, the ones making snRNAs, are for our health. They aren't just bystanders.
Right, so even though they don't make proteins themselves, they're essential for the protein-coding genes to work properly, which makes you wonder, how did they even find this RNU2-2 link, especially if it was hiding as a supposed pseudogene?
Well, that's where big data and clever analysis come in. They dug into the data from the UK's 100,000 Genomes project.
Okay, a massive data set.
Huge. They were looking for genetic links to neurodevelopmental abnormality, or NDA. But the key thing they did differently this time was they didn't filter out variants just because they had slightly lower CAD scores.
CAD scores, that's a prediction of how damaging a mutation might be.
Exactly. High score means likely problematic. They decided, let's look a bit lower down that scale at variants that might have been missed before. And boom, there was RNU2-2. It had been filtered out in the past.
So widening the net caught the fish, basically. What did they actually find in RNU2-2? Specific changes?
Yes, very specific changes. Recurrent de novo mutations.
De novo meaning new mutations, not inherited.
Correct. Spontaneous mutations happening in the child, but not the parents.
Yeah.
And they kept seeing the same mutations pop up at position 4 and position 35 of the gene.
The exact same spots.
The exact same spots. Initially in 9 people. Then, crucially, they replicated it, found the same mutations in another 16 cases.
Wow, 25 people in total, with the same new mutations at the same spots, all with similar issues. That sounds like strong evidence.
It's very compelling evidence for a direct causal link, absolutely.
And it wasn't just the genetics, right? You mentioned the initial 9 also had suspiciously similar symptoms.
Exactly right. That's what they call phenotypic homogeneity. These 9 individuals shared clinical features much more closely than you'd expect just by chance among a random group with unexplained NDAs.
Okay.
That really strengthened the case that this wasn't just a coincidence, but a distinct, recognizable disorder caused by these RNU2-2 mutations.
Makes sense. Okay, so we have the gene, the mutations, the link. Let's talk more about the gene itself. You mentioned it's similar to RNU2-1. How are they different?
Good point. They're very similar in sequence, but but their setup in the genome is different. RNU2-1 is the canonical one, the main workhorse. There are multiple copies of it clustered together on chromosome 17.
Like having lots of the same tool in the toolbox.
Kind of, yeah. But RNU2-2, it's a solo act. Just one copy sitting over on chromosome 11, unique sequence, unique location.
Only one copy. That seems vulnerable, maybe.
You could see it that way. What's really surprising, though, is that despite RNU2-1 having all those copies and RNU2-2 just the one, and being called a pseudogene previously, they found both genes are expressed at similar levels in blood.
Really? So RNU2-2 is definitely active, making its U22 RNA product, even with just one copy.
It seems so, which makes its role and the impact of mutations even more critical.
Okay, so let's get to the impact then. What does this RNU2-2 syndrome actually look like? If someone listening has a child with unexplained issues, what are the sort of hallmark features?
The paper lays out a pretty consistent set of core features. Things like intellectual disability, autistic behaviors are common.
Okay.
Also microcephaly, that's a smaller than typical head size, and hypertonia, which is low muscle tone.
Right.
And then two really prominent features, epilepsy and, interestingly, hyperventilation.
Epilepsy sounds like a major component. You said prominent.
Extremely. All 25 individuals in the study had severe, complex seizures, often starting really early, like in the first few months of life.
That sounds incredibly tough.
It is. And what makes it even harder is that the seizures were often pharmacoresistant, meaning difficult to control with standard medications.
Oh, wow. What kind of seizures?
A real mix. Spasms, tonic seizures, tonic-clonic, myoclonic absent seizures, a whole range. And in some cases, it progressed to what's known as Lennox-Gastaut syndrome.
Which is a particularly severe form, right?
Yes, very challenging. Characterized by multiple seizure types, developmental delay, specific EEG patterns.
Okay. And the hyperventilation, you said that was striking too.
It really was. When they looked at the specific symptoms using those HPO terms.
Human phenotype ontology terms, like standardized descriptions.
Exactly. Terms like seizures, microcephaly, generalized hypotonia, severe global developmental delay, and hyperventilation were significantly more common in the RNU2-2 group compared to other people with NDAs.
And hyperventilation specifically stood out?
Yes. It's not a term you see used that often in these contexts, and multiple independent doctors noted it for different patients with these mutations. It seems quite characteristic.
So that combination, the severe early drug-resistant epilepsy plus the hyperventilation, alongside the ID and microcephaly, could be a strong indicator for clinicians.
Potentially, yes. A cluster of signs to look out for.
Did the paper mention any physical features? Sometimes syndromes have subtle facial characteristics, dysmorphic features.
Yes, they did note some shared features among many affected individuals. Things like long palpebral fissures, the eye opening, sometimes with the lower lids turning out slightly.
Okay.
Also long eyelashes, a broad nasal root, large ears that might be set a bit low, a wide mouth, and teeth that were more widely spaced.
Subtle things, but a pattern maybe.
Exactly. No single feature is definitive, of course. But seeing several of these together with the neurological symptoms could add to the clinical suspicion for RNU2-2 syndrome.
Right, another piece of the diagnostic puzzle. So how does this RNU2-2 picture compare to the ReNU syndrome caused by RNU4-2 mutations? Are they easy to tell apart?
There is definitely overlap. You see seizures, microcephaly, hypotonia in both. But RNU2-2 syndrome does seem to carve out its own space. The developmental delays often appear more severe. The epilepsy tends to hit earlier and harder than typically seen in ReNU.
Oh, the hyperventilation difference.
And the hyperventilation, yes, that seems more specific to RNU2-2. Clinically, ReNU sometimes presents with different facial features, and the epilepsy might be later onset, maybe more focal, starting in one brain area. So related genes, yes, but distinct syndromes.
Okay, that distinction is important. Now let's dig into the mechanism. Why do these specific mutations, the ones at position 4 and 35 in RNU2-2, cause such problems? What do they actually do?
So those positions, 4 and 35, they're not just random spots. They're in a really critical region at the beginning of the gene, the 5' end.
Okay.
And what's telling is that this specific region shows unusually low genetic variation in the general population.
Meaning it doesn't usually tolerate changes well. It's conserved.
Exactly. Evolution tends to keep important functional regions stable. Low variation suggests it's doing something vital, and messing with it is likely bad news.
So what vital roles are position 4 and 35 involved in?
Well, position 4 in the U2-2 snRNA molecule itself, it's right in the part that needs to interact with another snRNA called U6.
Uh-huh.
That U2-U6 interaction is absolutely fundamental for the spliceosome's catalytic activity for the actual cutting and joining of RNA.
So mutation there messes up a key interaction within the splicing machine.
That's the idea. And position 35, that's part of a highly conserved sequence that's crucial for recognizing the branch site on the introns, those bits being removed.
The landing pad for the splicing machinery, sort of.
A critical recognition point, yeah. Getting that wrong means the spliceosome can't identify the intron boundaries correctly, so both mutations hit really crucial functional spots.
It sounds like throwing a wrench into delicate clockwork. Did they model how these mutations might affect things?
They did. They used computational modeling.
Yeah.
And the predictions suggest these mutations destabilize things.
Okay.
The position 4 mutation seems to weaken that crucial U2-U6 interaction, and the position 35 mutation likely messes up how well U2-2 binds to that branch site sequence on the pre-mRNA.
So the mutated U2-2 just doesn't work as well, basically. It's less efficient or less accurate.
That's the predicted outcome, yes. It impairs its ability to engage properly in the splicing process.
And these main mutations, the M.4GA and M.35AG, are they mostly inherited or mostly de novo?
They are germline mutations, so present in all cells from conception. And in most cases reported, they were indeed de novo new in the child.
Okay.
And interestingly, when they could trace the origin, most of these de novo mutations came from the mother's egg cell.
Maternal origin? Is that significant?
It's an observation. The reason isn't clear yet. But there's another twist with position 35. There's a different change, N.35AT.
Another mutation at the same spot.
Yes, but this one seems to be different. It looks like a recurring somatic mosaic variant.
Somatic mosaic, meaning it arises after conception and is only in some cells.
Exactly. Not in every cell like the germline ones. And interestingly, this AT variant was mostly found in older individuals.
Older individuals, why?
It seems consistent with something called clonal hematopoiesis. That's where, over time, certain blood cell populations with a particular mutation can expand. So this might be mutation that arises later in life, at least in blood cells.
Fascinating. So you have mutations present from birth causing severe neurodevelopmental issues and possibly another one at the same spot arising later. Complex.
It really adds another layer, doesn't it?
It does. So given U2-2 is so vital for splicing, the obvious question is, did they see loads of splicing errors in the patient's cell?
You'd think so, right. But that was actually one of the initially surprising findings. When they looked at blood samples, which are the most accessible tissue,
right,
they didn't find widespread splicing errors or missplicing, nor huge changes in overall gene expression patterns.
That seems weird. If a core splicing component is faulty, shouldn't splicing go haywire?
It does seem counterintuitive. And both the normal copy and the mutated copy of RNU2-2 were being expressed in the blood cells.
So why wouldn't they see the errors in blood? Any ideas?
The paper offers a few possibilities. One is that because this is likely a dominant disorder, one bad copy is enough. Maybe the normal RNA produced from the good copy is enough to keep things mostly okay in blood cells, kind of masking the defect.
Compensation, maybe?
Possibly. Another idea involves a cellular quality control system called NMD, nonsense-mediated decay.
The system that gets rid of faulty messenger RNAs.
Exactly. Maybe any badly spliced RNAs resulting from the mutated U2-2 get rapidly destroyed by NMD, so you don't see them accumulating.
So the errors are happening, but they're cleaned up quickly in blood.
That's a hypothesis. Or maybe there's some kind of dosage compensation happening specifically in blood cells, and it's not entirely clear yet from just looking at blood.
Okay, so blood might not be telling the whole story, especially for a neurodevelopmental disorder. Where do they suggest looking next?
Yeah, they emphasize that strongly. Future work really needs to look at more relevant tissues, specifically neuronal stem cells and the neurons themselves.
Makes sense. Go to the source of the problem.
Precisely. Since it's a brain development issue, you need to look at brain cells. And they also stress the need for animal models.
Like mice with these RNU2-2 mutations.
Exactly. That would allow researchers to really track how these mutations affect development and function across different tissues over time in a way you just can't do with patient samples alone.
Right, essential for understanding the downstream biology. Okay, stepping back again, the bigger picture.
Yeah.
You mentioned RNU2-2 mutations seem relatively common compared to other single gene causes of intellectual disability.
Yes. Based on the 100,000 genomes data, it emerged as a more frequent cause than most other known single-gene culprits they looked at. Still only about a fifth as common as ReNU Syndrome, but significant nonetheless.
So what does that mean for diagnosis? This must be important for families seeking answers.
Immensely important. This discovery provides a concrete genetic explanation for individuals with this specific constellation of symptoms, the ID, autism features, microcephaly, hypotonia, that severe epilepsy, the hyperventilation.
It gives a name to the condition.
Exactly. And that can end years of diagnostic searching, which is incredibly stressful for families. It provides answers, allows access to appropriate support, maybe informs management strategies, and opens the door eventually for targeted research into therapies.
That impact on families is huge. So let's try and synthesize this. If you were to boil down the key takeaways from this deep dive, what would they be?
Okay, I'd say. First, mutations in RNU2-2, a gene encoding a vital spliceosomal component, U22 snRNA, cause a distinct severe neurodevelopmental disorder.
Right.
Second, the core features are intellectual disability, autistic traits, microcephaly, hypotonia, and notably severe early onset, often treatment-resistant epilepsy, plus that risk of hyperventilation.
Okay, key features identified.
Third, specific recurrent de novo mutations at positions 4 and 35 seem to be the main drivers. And fourth, this was previously thought to be a pseudogene, highlighting we still have lots to learn about our genome.
Great summary. And despite finding the cause, there are still big questions about how it causes the disease.
Absolutely. We know the genetic trigger now, but the exact chain of events, how mutated U22 disrupts splicing in the relevant cells like neurons, and how that leads to the brain development problems, that's still largely unknown.
So that's where the future research needs to go, neuronal cells, animal models.
Exactly. Understanding the downstream cellular mechanisms is the next major hurdle.
So for you listening, what's the relevance here? Why should this matter?
Well, I think it shows the amazing progress being made in understanding the genetic basis of neurodevelopmental disorders. It really highlights how critical these intricate cellular machines like the spliceosome are. Even tiny parts matter hugely.
And the power of new technology finding these things.
Definitely. Advances in genomic sequencing and analysis are uncovering new conditions like this, offering hope for diagnosis, for answers, and maybe one day for treatments. And, the fact that a pseudogene turned out to be so important.
It's a bit mind-blowing.
It really is. It just underscores the complexity and the constant surprises hidden within our own DNA.
It's a fantastic reminder that biology is full of twists and turns. So maybe a final thought for you to ponder. Given this RNU2-2 story, how many other bits of our genome, currently labeled as junk or non-functional, might actually hold vital clues to health and disease?
It really makes you wonder, doesn't it?
It certainly does. This discovery opens up whole new avenues and gives us, I think, a much deeper appreciation for just how complex our cells really are. Thanks for taking this deep dive with us.